Hello, my name is Garth Kong, and today we're going to be talking about PyTOF and how that's being used to analyze CyTOF datasets. So first off, what is CyTOF? CyTOF is most similar to flow cytometry. In CyTOF, instead of using fluorescent reporters, CyTOF uses antibodies bound to rare earth elements that are then detected and quantified using mass spectrometry. And compared to flow cytometry, CyTOF can increase the maximal amount of features from 8, which is the maximum in flow cytometry, closer to 50 in mass cytometry. And now, current challenges with analyzing CyTOF datasets is that scientists are still using the gating strategy, which is, can you plot two protein features on an X and Y axis, and then select for distinct cell populations. However, by using the gating analysis method, you start to introduce a lot of selection biases, and sometimes gate boundaries can be subjective based on who's gating, as well as sample-to-sample variation. So ideally, we would like a method that is able to more holistically evaluate all the features at once. And in previous publications, most packages have been written in R. So in this project, we present PyTOF. It's written in Python using SnakeMake, and it's helping us to analyze CyTOF data in a more scalable way. So over for the methods, we have a series of tables. Each row is cells, that's our I, and each column is a feature, which is a protein feature, that's J. And then we replicate these cells by features matrices over your K samples. And the first step in PyTOF is that we normalize by time using Fink et al. 2013 paper. And what this means is that the longer your samples are in the CyTOF machine, the more skewed the signal is. But the skew is more or less linear. So Fink 2013 solves that for us. And then optionally, we can apply batch correction to our samples, and that's using SciCombine. We transform the data using the ArcSine transformation, followed by quality control. And this is where we get rid of the dead cells that don't really benefit us from the analysis. And then lastly, we can use other computational methods to help really analyze the data and put meaning behind it. So that means our dimension reduction techniques like PCA, t-SNE, and UMAP. Or if we want to look at more sophisticated models, we can use LDA or NMF. And the output of PyTOF is usually generic enough that you can build more classifiers or custom models on top of it as well. Okay, so we've built the PyTOF pipeline. We want to evaluate like whether it's working or not. So in the first dataset, what we have here is a healthy PBMC dataset. And PBMC is just a blood draw, peripheral blood mononuclear cells. In this dataset, the data comes from Stanford Blood Center, and there are 20 healthy patients. We apply PyTOF to the dataset, and after dimension reduction using UMAP, we see a separation of distinct cell types. Over on the left, we have the B cells, as shown with increased CD19 surface protein expression. And then we also see a really high CD56 protein expression, which corresponds to natural killer cells. Over for the bottom here, we see a large T cell population that are characterized by really high CD3 and CD8 and CD4 expression. And then up at the top, we have a CD14 positive monocyte population there. And these cells, using the same data set, this is visualized differently using a hierarchical clustering and a heat map. So here we have 5 total clusters. Each row is a cell, and each column is a protein feature. And over here in this cluster, we have B cells because there's really high CD19 expression just right here. And then these are the CD14 positive monocytes. These are the 2 large T cell populations. And then this is the natural killer cell cluster. And so with this data set, what we found is kind of a typical profile of a healthy donor. Now, if we apply this PyTOF pipeline to somebody with chronic lymphocytic leukemia, we see that, okay, so let me take a step back. So the samples were collected in Dr. Evan Lin's lab at Oregon Health and Science University. In this data set, there are 18 patients with chronic lymphocytic leukemia and 2 healthy patient samples. And after we apply CyTOF to it, we see that there are mainly 2 clusters. This data set is not as complex as the previous one. In fact, in one large bulb on the left, we see really high increase in CD19 and CD20 expression, which corresponds to a large B cell population. And this is known for this is what we would expect for somebody with CLL. And over on the right side, we have overexpression of CD3, CD4, and CD8 markers, which corresponds to immune cells. And over in the heat map, we see that there are like 4 clusters. 3 of them will be B cells due to the high expression of CD19. And then over here is the T cell population. So overall, what we have learned in this project is that, yes, CyTOF datasets can be analyzed using traditional single-cell bioinformatic methods like dimension reductions, heat maps, and in the PBMC dataset, we saw a strong separation of cell types that we would expect to see in peripheral blood. The B cells, the T cells, the natural killer cells, the monocytes. However, when we apply this pipeline to study patients with leukemia, we see that those cell populations are much more homogeneous, consisting of mainly B cells and T cells in CLL. So in conclusion, we believe that PyTOF was able to separate out the major cell types in CyTOF datasets. And in future work, we want to add on additional analytical modules to PyTOF to better process, to better analyze, and to interpret CyTOF datasets. Thank you so much. My name is Garth Kong. I work in the lab of Dr. Olga Nikolova at the Oregon Health and Science University. Thank you very much. Have a great conference.